Mm-hmm. Okay. All right. Sounds good. They keep these talks online? So that anybody can see them on the internet?
We can put a access password on.
All right, sounds good.
All right, let's get started. Today's seminar speaker is Charlie Brenner, coming to us from the University of Iowa. I know Charlie because the two of us overlapped briefly while we were both graduate students at Stanford. Charlie graduated, got his BS, I believe, in biology or biochemistry from Wesleyan University. And instead of going directly to graduate school, he actually worked for several years in the biotech industry, first with Chiron and then with DNAX, and did a lot of really nice molecular biology, but had decided by the end of that time that he really wanted to move into doing more enzymology. And so he came to graduate school at Stanford in the biochemistry department, and worked with Bob Fuller, studying the specificity of processing proteases, and did some very nice enzymology and protein biochemistry as a graduate student. And then sort of following that track, he decided to next pursue crystallography. So he went to Brandeis, where he worked with Dagmar Ringe and Greg Petzko as a Leukemia Society fellow. And there he sort of stumbled into this area of tumor suppressors that had sort of mysterious function, all of which seemed to have some bearing on nucleotide pools or sensing. And so that sort of became the focus of his work when he started his own lab at Thomas Jefferson Medical College and in the Kimmel Cancer Center there. And during his time at Thomas Jefferson, he rose through the ranks and eventually became the director of the Structural Biology and Bioinformatics program, won various prestigious young investigator prizes during that time. And then in 2003, he moved from Thomas Jefferson to Dartmouth, where he worked both in the Cancer Center there and in the departments of genetics and biochemistry as an associate professor and then a full professor. And then in 2009, he moved again to become the chair of the biochemistry department at the University of Iowa. And during that time as an independent investigator, his work has evolved from trying to understand the function of these tumor suppressor proteins to really understanding how nucleotide metabolism, and in particular NAD metabolism, really works, and how different precursors can actually influence nucleotide abundance and the function of NAD-dependent enzymes that turn out to be important in aging. And so today he's going to talk to us about, I should also mention this seminar is co-sponsored by the PSOC, which you should be very familiar with by now. Just get rid of that. Why is this?
Pretty close.
What's that?
Upper left.
It's not PowerPoint. Okay. So the title of this seminar is Counting Calories with NAD, New Biosynthetic Pathways for Life, Death, and Aging. Charlie.
Thank you, Jason. It's really great to be here at Northwestern. It's my first time here. I'm a big fan of the city of Chicago, and really appreciate the great people that you've set me up to talk to today. So as Jason said, the biological problem, one of the biological problems that we're trying to understand is calorie restriction, and how this relates to cellular aging. So this is a pretty well-known phenomenon that with fewer calories in a number of different model systems, there's a lower incidence of multiple diseases and extended lifespan. This has been observed for a long time, and the question is obviously molecularly, how does this work? So you need a genetically tractable system in order to make any headway into this type of problem. And Lenny Guarente forged ahead in the 1990s with the belief that you could use the yeast Saccharomyces cerevisiae to try to dissect this problem. And so there's two different models of aging in yeast. One is called chronological lifespan. The other is replicative lifespan. So chronological lifespan is how long do cells survive when they're grown to stationary phase, how much time, right? And replicative lifespan is how many times will a mother cell produce a daughter. Because it turns out mothers will only produce a certain number of daughters. And they'll produce, in this particular strain background, about 20 daughters, but about 40 daughters if you calorie restrict them. You calorie restrict a yeast cell by growing it in 0.5% glucose rather than in 2% glucose. Now, in this type of a screening regimen, one can find a few long-lived mutants, one can find a lot of short-lived mutants, and then one can find an interesting class of mutants that are both short-lived and don't respond to calorie restriction. And that's one of the hallmarks of the SIR2 mutation that was reported by Guarenti. Now, even SIR2 mutants are not short-lived in the chronological lifespan paradigm, and there's some strain backgrounds in which they're not even short-lived in this assay. But in a wild-type assay, a SIR2 mutant has a short lifespan, and you can't extend its lifespan by reducing the glucose. So the question is, how does that work? So the short answer that appeared to emerge from the Guarenti laboratory has to do with the requirement of the SIR2 gene product and its family members, which are called sirtuins, as NAD-dependent enzymes. And the speculation was that in some way, NAD, as being essentially a substrate in glucose utilization and glycolysis, as well as a physical substrate of NAD-dependent histone deacetylases, might in fact be the metabolite that is altered when cells are restricted. And that would increase SIR2 function. And as you may know, if you're a Saccharomyces cerevisiae aficionado, one of the things that happens in the mother cell is accumulation of ribosomal DNA circles. And SIR2 has a role in repressing recombination, repressing formation of those circles. But even if this is true, and I would argue that it's oversimplified, we would need to understand the nature of NAD metabolism that is altered. And there, with very little data, I would say, the Guarenti lab came up with the idea that the NAD plus to NADH ratio might be modulated by glucose. David Sinclair cloned an enzyme involved in nicotinamide salvage and said that fundamentally what happens in glucose restriction is reduced nicotinamide metabolite. And then we showed through some of our experiments that we did that because one can extend lifespan with increased net NAD synthesis, that potentially increased net NAD synthesis might be the mechanism by which calorie restriction alters NAD metabolism and thereby SIR2 function. Turns out, I think today I have enough data to say that none of these models are correct. But we're going to show you what we've done in the last few years to identify genes and pathways in NAD metabolism and find all the players that are potentially modulable, if that's a word. So before I do that, I'm going to take you back, I guess, 100 years, or 105 years by now, in the history of biochemistry, in the history of the molecule that we call NAD. And it was really first described by Arthur Hardin around 1905. Now, this is really the ancient days of biochemistry. No one here alive remembers them, but it's such an ancient day that glycolysis is the coolest thing that you could study, and that what we're trying to do here is find the one error, the one significant error that Louis Pasteur ever made in his life, which was believing in vitalism. And that was essentially disproved by the invention of a very high-tech instrument by the Buchner brothers. I refer, of course, to the sintered glass funnel that was required in order to make a cell-free extract that could do glycolysis. Now, why did Hardin's glycolysis work better than everybody else's? Why do we remember Hardin? Because Hardin basically, and his student von Euler, basically did a little fractionation. And they got basically a bag of enzymes, a bag of proteins, and they had a bag of small molecule, or they had the dialyzable stuff, the heat-stable dialyzable stuff, and they called the bag of enzymes zymase, and they called the dialyzable heat-stable stuff cozymase. And then when they added cozymase back together with zymase, they could get cell-free glycolysis. That was great work, and there's actually 2 Nobel Prizes for that. First for cozymase, essentially, and then for starting to fractionate those enzymes, those glycolytic enzymes. So this, was hot and sexy, and so Otto Warburg, a little bit later, actually figured out what cozymase was. So cozymase was really 3 different things. It was ATP, magnesium, and something that they called at the time DPN, or diphosphopiridine nucleotide. So diphosphopiridine nucleotide, we call that NAD, or nicotinamide. That's a nicotinamide base. Sorry, that's a nicotinamide, reduced nicotinamide base. That's an adenine, and then it's the dinucleotide of those 2 mononucleotides. And what Warburg understood by the middle of the 1930s is that cozymase has an enzyme-like coenzymatic function because this is not a destructive process, right? Because in the interconversion of an alcohol to a ketone or aldehyde, the NAD, the nicotinamide base, is the hydride group acceptor, producing this reduced form of NAD, which we call NADH. So it's not a destructive process. You can go back and forth between NAD plus NADH as many times as you want. And so this was the really hot thing, because now you had to figure out how does a cell make NAD, and nutritional biochemistry was the thing that one did out here at a Big Ten university. And so Conrad Elvehem at Madison went around and his staff finding dogs in which they could perform the following experiment. Starve the dogs to the point of near death, at the point at which they have black spots on their tongues, and find non-protein factors, and I see people are very upset by this description, and I am too. Find non-protein fractions from liver that will make the black spots go away. So that would be some vital nutritional factor that was limited by the process of starving these dogs. And essentially, Elvehem and his co-workers identified 2 different chemical entities, very closely related chemical entities that did that. We call them niacin. Okay, chemically, they're nicotinate or nicotinic acid, and the amide form of that, nicotinamide. And he actually, at the time, didn't even know that they were DPN or NAD precursors, but in a few years, they figured that out. That basically nicotinic acid, these anti-black tongue factors were the precursors, the vitamin precursors of NAD. And that the cell also has an endogenous de novo pathway by which the cell makes NAD. And so a couple of vocabulary terms. These are vitamins, and because you can see this nicotinamide base in NAD, the pathway by which vitamins are utilized is called salvage. And then this, if you were to draw a tryptophan, you wouldn't be able to find the carbons and nitrogens in tryptophan that go on to NAD, because there's 8 steps and there's a lot of rearrangements. So this is de novo. This is making NAD from scratch. So now you might also know that there are some drug-like properties of these 2 compounds, and the drug-like properties of those 2 compounds are not identical to each other. The side effects are different, And so nicotinic acid is the most useful compound known to man and woman to control dyslipidemia at high doses. 15 milligrams a day will prevent pellagra, which is a human nutritional condition that is related to tryptophan deficiency. But a gram a day will increase your HDL cholesterol and lower your LDL and free fatty acids. High-dose nicotinamide can be useful for stroke, but this turns out to inhibit the sirtuins that we're going to be talking about in a minute. So now we've got to figure out the enzymes, and Arthur Kornberg was the first person to try to crack this problem. Is before this picture was taken, because he was already focusing, obviously, on DNA replication. But back in 1948 at Wash U, he actually purified the first NAD biosynthetic enzyme. And it's basically what we would call NMN adenylyl transferase. So it takes the mononucleotide form of NAD and in ATP-dependent fashion turns it into the dinucleotide. And that's a reversible enzyme. So then, very quickly, finishing the history of NAD, for those first year or second year graduate students in the audience, when Jack Preece walked into Phil Handler's office in the middle 1950s at Duke University, and this is, I think, the head of the National Academy of Sciences and a very distinguished biochemist. The young Jack Preece didn't look like that at the time. Said to Phil, what do you think would be a good project for me to do in the next 3-4 years? And Handler said to Preece, why don't you go in the lab and why don't you find every enzyme and every biosynthetic intermediate between nicotinic acid and NAD. And by the way, I want you to see if you can do that for nicotinamide as well. And he actually did that in a couple of years. He got the enzymes from his own blood and from yeast. And they're the same enzymes, and they go through these steps. I won't go through the enzymology, but you can think of it as Preece-Handler's first, second, and third enzyme. And basically, the first one takes a base and makes a mononucleotide. The second enzyme is very similar to Arthur Kornberg's enzyme. It takes a mononucleotide, NAMN in this case, and turns into a dinucleotide. And the third enzyme turned out to be Charlie Brenner's favorite enzyme 45 years later. The third enzyme turned out to be something called glutamine-dependent NAD synthetase. Okay, so now this thing is all solved. It's not interesting anymore. This got put into your textbook in the late 1950s, early 1960s in this form. So tryptophan goes to NAD in 8 steps. Preece-Handler in 3 steps. Notice that they both collide right there. So that's cool. So Preece-Handler's second and third enzyme are the 7th and 8th enzyme of the de novo pathway. You already know the de novo because I taught you the vocabulary words. NAD, NADH is interconvertible. NADP, NADPH is interconvertible. It's a little complicated around there. But then there's something else that was known in the 60s, which is there's some destructive pathway that breaks down the NAD to nicotinamide. So why would, after all 8 steps and prevention of black tongue and pellagra from the vitamins, this is obviously good stuff, right? This is required for glycolysis. Why would the cell break down NAD into nicotinamide? And then further, how do you recycle nicotinamide? We already know this vitamin stuff. But what are the remaining problems? I mean, why does the cell break this down? What's that enzyme? Why destroy the NAD? How do you salvage this enzyme? And you're all thinking, well, dummy, draw an arrow from right to left, and you've got it done. And it turns out fungi do it that way, and a lot of bacteria, including E. coli, do it that way. But what's true neither in E. coli and fungi is not true for the elephant, and the elephant doesn't do it that way. So humans do not have that arrow, except in our gut where there are bacteria. And then, of course, what are the genes that encode these enzymes? But this is the 1960s, and so as molecular biology was developed and became possible to identify those enzymes. People did it on interesting things like phage morphogenesis, right? And so you go all the way to the year 2000, and you take the world's most boring protein, histone. Is Jonathan here? And you take the most, too ubiquitous to be regulatory, right? That's what we thought in the year 2000. And you take the world's most boring and abundant small molecule, NAD, 1 millimolar concentration in cells, too abundant to be regulatory. And sort of what happens when you mix NAD and nicotinamide and you discover reversible protein lysine acetylation. Some of that doesn't require NAD, but some does. So histone acetylation, which is largely associated with activation of genes, doesn't require NAD, but one class of deacetylases requires NAD. And so NAD-dependent lysine deacetylation represses recombination, silences transcription, and also modifies the function of a lot of enzymes, a lot of metabolic enzymes, as a matter of fact. And so this was discovered by not only Lenny Guarente, but other people as well, that in this reaction of the type 3 lysine deacetylases, I'll occasionally call them HDACs or histine deacetylases, but the substrates are not always histones. There's an acetylated lysine, which is all the chromatin people are thinking about, is the acetylated lysine, and that's going to lysine, right? But turns out SIR2 won't do that without another substrate, and the other substrate is NAD. The product of that reaction, in addition to the deacetylated lysine, is an acetylated ADP ribose, a broken NAD, where the ADP ribose moiety picks up the acetyl group and releases nicotinamide. So, and this reaction is required for gene silencing and lifespans in response to calorie restriction. So this sounds like, some pretty interesting cell biology. And so that indicated that these NAD glycohydrolases, these mysterious enzymes that broke the NAD down to nicotinamide, that were known about 50s and 60s, these NAD glycohydrolases turn out to be sirtuins. And they also turn out to be enzymes like poly-ADP ribose polymerase, mono-ADP ribose transferases, cyclic ADP ribose synthases. Any enzyme that you can think of that has an ADP ribosyl product that cyclizes ADP ribose, that transfers ADP ribose to a polymer or as a post-translational modification, all of those enzymes get the ADP ribose from breaking NAD. Essentially irreversible reaction. So these NAD-consuming enzymes are those three things. So we happened, for reasons too numerous to describe, be working on this enzyme, glutamine-dependent NAD synthetase. I kind of liked it because it had 2 active sites, and one of the things was sort of related to our tumor suppressor story. But we were trying to figure out how this enzyme works, glutamine-dependent NAD synthetase. And we'd made mutants in each of the active sites and just tried to see if we could make cross-complementing mutants and so forth. The cool thing was that if you look at the pathway, and a number of molecular biologists that were practicing biochemistry without a license looked in the yeast genome at all of those NAD biosynthetic enzymes and assigned gene names to them, right? And and so you can see the de novo pathway, which I always draw coming from the left, goes to NAD through QNS1. Nicotinate goes through QNS1, and in yeast, but not in humans. In yeast, nicotinamide also goes through QNS1. So QNS1 should be required for life under all circumstances, no matter what. We realized that, and people started manipulating these enzymes because Sinclair showed hyper expression of PNC1 will extend lifespan, in support of his idea that low nicotinamide is the means by which calorie restriction works. So we thought, I don't even know if this pathway is right. I mean, no geneticist has actually messed with it. And so, for example, this enzyme, Priest-Handler's first enzyme, if this enzyme, NPT1, can utilize nicotinamide as a substrate, it's not going to make NAMN. It's going to make a compound called nicotinamide mononucleotide, which was Arthur Kornberg's substrate, which is not even on here, not even on this diagram. And that enzyme, which Handler purified, that enzyme might actually use that substrate. And then moreover, my friend Vern Schramm can get this enzyme to go backwards. He can give SIR2 nicotinamide and acetylated ADP ribose, and he can get SIR2 to make NAD, right? So if that's the case, then there's a number of ways in which I might, if I use genetics, I can determine whether this pathway is correct and complete. So for example, the first thing is see whether nicotinamide can bypass this gene, and then see whether anything else can bypass this gene. So the answer is nicotinamide cannot bypass. So this enzyme really is totally specific for that substrate. SIR2 can't go backwards, but nicotinamide riboside, which is a compound that was known to be an NAD precursor in bacteria, but was not thought to be an NAD precursor in eukaryotes, will actually bypass. So there's got to be another way to do this. There are nucleosides, so nicotinamide riboside is basically the nucleoside precursor of NAD, right? So you need a nucleoside kinase. I'm looking for an enzyme that probably in an ATP-dependent fashion will make NMN, right? And then I can go and look up in the literature whether Arthur Kornberg purified his NMN adenylyl transferase from yeast as well as potatoes, which I think he did. I'm not exactly sure, but it wouldn't surprise me, right? So I'm looking for an enzyme that will phosphorylate a nucleoside. And if you look in the previous figure, we looked at all known nucleoside kinases, and they didn't. When you knock them out, you could still utilize nicotinamide riboside. So we had to use a biochemical genomics approach to purify the enzyme that encodes this activity. And we found a single enzyme in the yeast proteome that had this activity of phosphorylating NR. But of course, that doesn't prove that it's really nicotinamide riboside kinase. Only genetics does that. So you knock out NRK1, and now in the QNS1 background, nicotinamide riboside cannot provide for viability. And then we find nicotinamide riboside kinase in humans as a NRK1, NRK2. Those are interesting genes, by the way. So then Pavel Vyagnovsky, in an experiment that did not require the use of any starving dogs, showed that that milk is a source of nicotinamide riboside, and much better than the dog experiments, which would have been positive if you gave the cells enough tryptophan, or nicotinamide riboside for that matter. Here, in a pathway-specific manner, we can show that they're using something in the whey fraction of milk is actually a source of nicotinamide riboside. So now we get a little bit more information about NAD biosynthesis, right? And so we basically add, the single arrows means vertebrates or fungi. Double arrows means that vertebrates and fungi do it the same way. So now we've basically added 2 precursors and a new gene. But we don't yet know whether there are any additional arrows to or from nicotinamide riboside. Nicotinamide and nicotinic acid are metabolites in addition to vitamins. So a vitamin is something that comes in from the outside, which is what I've been using nicotinamide riboside for. A metabolite is something that the cell makes. And it could be both. And then, of course, we want to know whether there's any role in silencing or anti-aging. And the answer to all of these questions is yes. So initially, we thought, boy, these experiments are going to be really, really easy, because nicotinamide riboside did everything that we asked it to do. Improved SIR2-dependent genome stability. I won't go through the data, but the red sectors in this colony color sectoring assay are indications of high recombination You could do gene silencing assays in which nicotinamide riboside represses gene silencing. You do NAD biosynthetic assays, and you get more NAD per cell when you give the cell nicotinamide riboside. And a lifespan assay, you shift this death curve to the right, so you prolong lifespan. So it was doing everything, right? And then we did a very simple experiment, and we said, well, let's knock out the NRK1 gene and better see that nicotinamide riboside fails to be an NAD precursor when you knock out NRK1, right? Because I just showed you, you need the NRK1 gene for NRK1, for nicotinamide riboside to bypass those other pathways. So we got a surprising result. So here's the wild type. There's almost 1 millimolar NAD by this assay in cells grown on vitamin-free media. And you give them, this is intracellular millimolar, and I can explain how I do that if you want. You add nicotinamide riboside to a wild-type cell, and you basically double the NAD. There's 2 millimolar. These cells also live longer. Now you take these NRK1 mutants, they have the same thing. You give them nicotinamide riboside, and you get a millimolar more of intracellular NAD. So this seemed like a bad day in the lab, right? So we thought, well, we've shown that NRK1 is required for nicotinamide riboside to bypass QNS1. But maybe nicotinamide riboside has another way of going. So that's what my student, Peter Belenke, figured out. It had another way of going, and probably draw an arrow there. So that's what he figured out, is that if you knock out nicotinamidase with NRK1, then nicotinamide riboside is incapable of being utilized. And then it was just a process of figuring out what are the genes and enzymes. So nucleosides can get broken down to bases 2 ways. One requires water, the other requires a phosphate. You look in the yeast genome, there's a phosphorylase. It's a famous enzyme. It's called purine nucleoside phosphorylase. You look in there's also something else called methylthioadenosine phosphorylase. Then there's an enzyme called URH1 for uridine hydrolase. And it turns out that although those things have gene names that associate one of them with purine metabolism, and that associate the other one with pyrimidine metabolism, those gene products actually play roles in NAD metabolism. In fact, our biochemical and genetic data indicate that URH1 is really a misnomer. It's really nicotinamide riboside hydrolase. It favors nicotinamide riboside 100-fold over the previously known thing. So, and they have non-equal roles. We quantitated these things. You put PNP1 back in, you get some effect. You put URH1, you get more. Peter was happy when he did that experiment. So, and then the nice thing, the kicker, was that nicotinamide riboside really does extend yeast lifespan with respect to growth in vitamin-free media, and by a lot. So in vitamin-free media, wild-type cells in our strain background only produce about 12 to 13 daughter cells. A mother only produces 12 to 13 daughter cells. And if you give them nicotinamide riboside, then you get about 10 more daughter cells per mother. You delete either one of these pathways, the NRK pathway or the URH-PNP pathway, you get about half of the effect. And then there's this other gene called MEU1. There's a very small effect. If you delete all of those nicotinamide riboside salvage pathways, the NRK pathway or the other pathway, then nicotinamide riboside is powerless to extend lifespan. So it's a simple mechanism. Unlike a lot of the mechanisms of lifespan extension, this is just making more NAD. Now, whether or not this is a mechanism of calorie restriction, or whether this is just something that Peter Belenke and Charlie Brenner did in the laboratory, that's another matter. And we'll get to that very shortly. So, but first, let's finish the metabolites. So the phone rang one day, and Haewon Park at the University of Toronto had solved a crystal structure at the Structural Genomics Consortium. And they were just blazing through the human proteome for soluble enzymes. So NRK1 was one of their, quote, targets. And you know, he called me up and he said, it looks like a metabolite kinase. And I said, yeah, I know it looks like a metabolite kinase. It has, you know, 30% similarity to uridine cytosine kinase. But the only thing that's going to be interesting about that, and it was fortunate, actually, it was fortunate that I was a grad student when Al Edwards, who's one of the directors there, was a postdoc, because what they normally do in Toronto is they just deposit. They just deposit all the structures that they solve. Because the charity that supports their consortium believes in open access and, you know, just you distribute the coordinates. But the coordinates have no value unless somebody does an experiment with them, right? And I argued, it didn't take much argument, that what we really need to do is we need to understand how the structure of this enzyme mediates its specificity. I thought its unique specificity for nicotinamide riboside, right? So what does nicotinamide riboside look like, right? It's got a plain vanilla ribose, right, with a 2, 3, and 5 oxygen, but every nucleoside has that, right? So adenosine kinase probably has the same 3 contacts for the 2, 3, and 5 oxygens. So what's specific about a nicotinamide riboside kinase? Well, it's got to be looking at the nicotinamide base. All right? So what's special about nicotinamide base? Well, it's a non-substituted pyridine, not pyrimidine. It's a non-substituted pyridine base except for this carboxamide group, right? So I said naively to Heywan, it's going to have something. It's going to exclude the phosphorylation of UMP and CMP by some mechanism, by, you know, not allowing substituents there and there. And then it's probably going to have a way to recognize the carbonyl oxygen from the amide group, from the amino group, right? And he said, well, all I have is, you know, ADP in my crystal structure. I said, okay, well, I'll give you the nicotinamide riboside and then something called thiazofurin that we won't talk about. And we'll see how this is recognized. So it turns out that in terms of discriminating against UMP-CMP, everybody's right. So sterically disfavors those positions. But in terms of the enzyme recognizing the carbonyl oxygen from the amino group, you could turn this thing around in the model 180 degrees, and the enzyme's not going to see that at all. It's just geometrically complementary. There's no electrostatics that would be able to tell them apart. So we said, heck, I wonder if this enzyme would phosphorylate that. And if it phosphorylates this compound, nicotinic acid riboside, which forget bacteria, which bacteria had nicotinamide riboside, so our invention, as it were, is not a novel composition. It's a novel vitamin to humans, but there are bacteria like Haemophilus influenza that use nicotinamide riboside. That was known for a long time. We just drew this compound on the napkin and said, I wonder if nicotinic acid riboside is a substrate and potentially a metabolite. And it turns out it is. So nicotinamide riboside kinase 1 and 2 both phosphorylate nicotinamide riboside, nicotinic acid riboside with essentially equal kinetics. And nicotinic acid riboside can be utilized by cells. There's a vitamin transporter for nicotinamide riboside that Peter Belenky also identified by screening. We don't think that there is a transporter for nicotinic acid riboside. But this is actually a pretty specific molecule. Yeast really wants nicotinamide riboside. It's proton dependent. It's very specific, and so forth and so on. 10 transmembranes. So the last issue that we had to figure out in terms of metabolism before we attacked calorie restriction is, do cells make these compounds endogenously? Clearly, there's a transporter for nicotinamide riboside. And And so it's looking for nicotinamide riboside from outside of the cells, but is there a function for the NRK pathway or for this other nicotinamide riboside salvage pathway when you don't give cells any nicotinamide riboside? And from the NAD levels in the cells, we thought probably yes, because this is intracellular NAD, and this is optical density. So it turns out as you grow a yeast cell culture, the NAD level per cell goes down. But if you delete the URA, you delete both of the pathways. You don't need to know their names. You delete both of the nicotinamide riboside salvage pathways. The amount of NAD per cell is lower. And these are not under conditions in which the cells are getting nicotinamide riboside. So it suggests that the cell is just like the cell makes nicotinamide endogenously by breaking down NAD. In some way, the cell must be making nicotinamide riboside and or nicotinic acid riboside. So we needed mass spec. So we collaborated with Bob Kennedy at University of Michigan initially. My student, Katrina Bogan, and Bob Kennedy developed this metabolomic assay in which they used hydrophobic interaction liquid chromatography to separate the NAD metabolome, plus a few other compounds we were interested in, and then use LCMS on a triple quad mass spec in order to measure the concentrations of metabolites. And to our delight, wild type cells have nicotinamide riboside, and they also have nicotinic acid riboside. So there's an amount of these compounds in cells in the micromolar. Now, when you delete both of the pathways that utilize nicotinamide riboside from exogenous sources, so these are the genes that are required for the longevity benefit, for example, of nicotinamide riboside, delete those pathways, the metabolite levels go up. So now the cells accumulate these nucleosides. And then, so Katrina, boldened by this, said, okay, I think I can now do a biochemical genomic screen to identify the phosphatase, essentially, the 5' nucleotidase that will presumably take this phosphate group off of NMN and make the nucleoside. So she identifies 3 gene products that can do that and makes a bunch of double mutants. One is called ISN1, the other is called SDT1. Here's a lot of data on this slide. And essentially, it indicates that when you make a double mutant of these 2 enzymes that biochemically have the property of dephosphorylating NMN, NAMN, you knock out both of them, and now you don't get accumulation of the nucleoside. So that's essentially genetic evidence that enzymes that biochemically will dephosphorylate the mononucleotides, in vivo, they have that role. So that's Katrina. And so we've discovered increasing complexity in NAD biosynthesis with the identification of these various pathways. Question is, what happens when you calorie restrict cells? And so there's 2 different ways that we can extend lifespan. One is by adding vitamins. So you compare vitamin-free media to these 2 conditions to their immediate right. And then the other is glucose restriction. And so we decided to take this experiment and measure all the NAD metabolites and see whether calorie restricting cells does the same thing as adding a vitamin. And in fact, does calorie restriction do anything to the NAD metabolome? And this is really a good control because vitamin addition clearly increases net NAD biosynthesis. So nicotinamide, NR, NAR, NMN, NAMN, NAD, NADH, all of those things go up. They go up from they go up 10 to 20-fold, most of them. NAD only goes up 2-fold. But a lot of these, no, they don't go up 10-fold. Well, a couple of them go up 10-fold. They go up very substantially. And it's not only one NAD metabolite that goes up, but virtually all NAD metabolites go up. And it's very few other metabolites go up when you add an NAD precursor. Now, when you glucose restrict cells, and notice here, I want you to notice nicotinamide. So we can measure an intracellular concentration of nicotinamide at 2 micromolar. And NAD precursor addition, nicotinic acid in this case, will increase the nicotinamide level by 15-fold, actually. And so that means that in this YPD condition, I'm capable, or my assay is capable of seeing a reduction in nicotinamide levels if nicotinamide levels, in fact, go down. But when you calorie restrict cells and you go from 2% glucose to 0.5% glucose to 0.2% glucose, nicotinamide levels don't go down, contrary to the Sinclair prediction. And NAD levels don't go up, and NAD to NADH levels don't strikingly change either. Now, some of the enzymes that we've discovered, however, including ISN1, which is one of the NMN nucleotidase, turns out to be regulated at the protein level, both by nicotinic acid and by glucose. And so this is kind of overexposed, I'm afraid. But I think you can see the data here, that at low nicotinate, there's low ISN1, and at higher nicotinate, there's high ISN1. It turns out you get this result even when cells are incapable of using nicotinic acid. You get this result even in an NPT1 mutant. You get this result even with benzoic acid, which is a non-metabolizable form of nicotinic acid. And glucose, it turns out, regulates ISN1 quite strongly as well. So glucose restriction actually reduces the amount of ISN1. So we are seeing the effects of both. So metabolites like nicotinic acid and glucose aren't changing the abundance of metabolites, but they're changing the abundance of some of the NAD biosynthetic enzymes, and in particular in this nicotinamide riboside salvage pathway that we work on. SDT1 and FEM8, which are two of the other things that Katrina discovered, turn out to be in the Pho pathway as well as in the NAD pathway. Remember, think about the enzyme. It's a 5' nucleotidase, so it's taking a phosphate off of a nucleotide and making a nucleoside. It turns out that this is a part of a whole class of enzymes that are upregulated when you phosphate deplete cells. And I think I might have some phosphate depletion data here. This is, I guess, when you grow cells to stationary phase, but you can also show the same thing when you phosphate deplete data. When you delete Pho85, you also increase, it turns out, transcription of FEM8, and that depends upon the Pho4 pathway as expected. So ISN1 turns out to be positively regulated by nicotinate, negatively regulated by glucose. SDT1 and FEM8 turned out to be in the phosphate pathway, and so we've got some work to do to dissect how these different metabolic pathways are intersecting with each other. We have a ways to go before we actually understand how calorie restriction works, even in the yeast system, now that we've identified all the players. But the simplistic models that were formulated with an incomplete set of genes and enzymes, I think I'm confident enough to say are probably incorrect. So I think at this point, I'm just going to sum up and show you that we've identified two salvage pathways from nicotinamide riboside and nicotinic acid riboside. We've identified for nicotinamide riboside a dedicated transporter. We found that both of these compounds are both vitamins, have both vitamin properties as well as endogenous metabolite properties. The metabolite properties are produced by 5' nucleotidases that were identified in my lab by Katrina Bogan. And they're regulated post-transcriptionally by vitamins and by glucose, by vitamins and glucose, and transcriptionally by the Pho system. Vitamins increase net NAD synthesis, increasing lifespan, and that has tight SIR2 dependence, But I do not claim that this is calorie restriction mimetic. And parenthetically, I would say the big problem in this field is that a lot of the individual results are fine. But it's when somebody tries to claim that it explains the French paradox or how calorie restriction works in a mouse from a yeast experiment, or that you can explain, every aspect of calorie restriction through one gene product like SIR2. That's where people really get in trouble. And then finally, from the metabolomic that work that we're doing, we've identified some targets of calorie restriction that include IMP, actually, and NAMN metabolism, both of which are mediated by ISN1. And we've seen some crosstalk with the Pho system. I think I wrote this talk when I was going to UT Southwestern, where they know a gentleman named Andre Osterman very well. So Andre and I have another metabolite that we have found from a broader metabolomic screen that actually is induced by calorie restriction and specifically depends upon SIR2. And so I think I'll stop and I'll take your questions there.
So you showed us that NR provides lifespan extension.
Yeah.
Does NAR provide a similar lifespan extension? Is that also true in organisms other than?
Okay, good question. So NAR is not likely to work as well as NR because it doesn't have a specific transporter. So pound for pound, it's not going to work as well. But whether it works at all, I haven't determined. Our simple mechanism would predict, but it's only a prediction, that it would extend to the degree that it can elevate NAD, because that's what seems to be activating SIR2 in this system. Now, the vertebrate system is very interesting, of course, and the best results actually come from Jeff Milbrandt at Washington University, and these are not organismal experiments, but they're experiments in which you're looking at neurons to survive axonopathy. And it's pretty well known that there's a WLDS mouse that overexpresses an NAD biosynthetic enzyme. Actually, WLDS encodes NMN, adenylyl transferase, of all things. And when Milbrandt wanted to see what metabolites would allow a neuron to survive axonopathy, he discovered that nicotinamide riboside is the only NAD precursor that will allow a neuron to survive axonopathy without simultaneous gene therapy. So if he gives a neuron nicotinic acid, he's got to re-express Priest-Handler's first enzyme. If he gives it for it to work, because it doesn't express that pathway, if he gives it nicotinamide, he's got to express an enzyme called NAMPT. But just the process of damaging the neuron actually induces expression of NRK2. So NRK2 appears to be a damage-responsive transcript, such that in certain tissues, nicotinamide riboside is protective. And so we're really, really interested in the vertebrate translational implications of that.
One other question. Is it bulk levels of NAD, or is there some special population of NAD molecules that actually is modulatory to these pathways?
It's entirely possible. There's three current schools of thought on that. One says it's all about the mitochondrial pool. One says it's all about the nuclear pool. And then there's me, who hasn't figured out a way to separately measure those pools. And so I've not joined any camp. Yeah.
So do you think SIR2 can act in reverse in because the SIR2 reaction is actually reversible?
Yeah. I don't think so. No.
Let's thank Charlie one more time.